Okay, so here's my lab visuals from the DNA extraction of primarily animal blood cells as well as plant cells. For the blood cells themselves, here we have the setup of the first sample. So we had collected 5 milliliters of pure animal blood and put into the large centrifuge tube, and there's a lysis buffer added. This was centrifuged for about 5 minutes. The supernatant was collected, and then another lysis buffer was added to the pellet of white blood cells. This was also centrifuged to kind of fully condense the pellet. So here you can see another visual of the separation in a microcentrifuge tube. So you have the pellet, the solid condensation. It's a little bit more dense here at the bottom, and then the supernatant that floats above, and that was just extracted using a micropipette. Following the addition of the white blood cell buffer, you can see here there was cold chloroform was added to the sample to separate the white blood cell DNA. The suspended and solid DNA was then collected using a capillary tube here. So what we had done was the DNA was sitting in this beaker, and it was able to be seen. It was solid, kind of like all throughout the beaker. So what we had done is placed the tip of the capillary tube into the center and twirled it, and the DNA kind of bound onto itself as well as onto the capillary tube, allowing us to pull the solid completely out. It was then allowed to dry and then placed into a microcentrifuge tube with more buffers to dissolve the DNA and give us a liquid. This process was duplicated. This was duplicated doing strawberries. This process was almost identical, just used different buffer types. The purpose of this was to be able to compare the DNA amount between white blood cells and strawberries, as well as the concentration in each sample, being that strawberries have more chromosomes than animal blood. So in conclusion here, there's a little visual of a white blood cell, but the white blood cell is where we were collecting the DNA from. As you can see here, it's more complex than a red blood cell, so it should contain more DNA in theory, making it easier to get a valid reading. The results are going to be measured in a qubit, and the actual DNA amounts itself are going to be recorded and graphed for a visual between both samples to see what has the higher amount of DNA and if there are potential errors in the process of extraction. So that will be in a follow-up video, but the procedure for the animal blood was pretty straightforward, as well as the plants.